For 6 days, Research Vessel Eastward has been at sea. She sailed from Duke University's Marine Lab at Beaufort with a team of oceanographers to make a detailed study of southern coastal waters. The scientists have been measuring water depth and temperature, currents, salinity, oxygen content, and studying the tiny organisms that make up the bottom links of the food chain. Eastward's course for this cruise is a systematic zigzag pattern along the coasts of South Carolina, Georgia, and Florida. There are stops at carefully selected stations that stretch from shallow waters near the coast to the deep waters off the edge of the continental shelf. Cruises like this are being made at each season of the year, collecting data which over a period of time can be assembled to provide a better understanding of how the ocean supports its many forms of life. One important use for the periodic research data Eastward is gathering is that it can be used to detect changes in the marine environment. Not only changes that occur with the seasons and the weather, but also changes caused by man. One member of the scientific party, Elizabeth Waiters, is interested in how man-made pollution affects water pollution. She samples the air and the water, comparing the amounts of heavy metals in both.
Well, this is an air sampler, and we have it on the bow of the ship behind the anchor hoist to protect it from, really bad weather. It pulls in the air at 2 liters per minute through a solution of permanganate and sulfuric acid. It traps the mercury, which may be in the air, into that solution. We take it, we run it for 8 hours and 20 minutes, and that gives us roughly about 4 or 5 stations on a transect. Well, we take it back to the office and we analyze it for the content of mercury that may be in the air as in relationship to the mercury that we sample, in the water.
Detecting small quantities of metals in the water requires careful use of uncontaminated equipment. A long pole fitted with plastic tubing serves to collect the water. The sample is taken a little distance from the ship to avoid water near the metal hull. Water is pumped through the plastic tube into the wet lab. There it is collected and stored in plastic containers. Later, the water will be analyzed in specially equipped labs on shore. Betty Brandow is collecting samples to be analyzed for radioactive isotopes, like those produced by nuclear reactors. The scientists wonder if nuclear pollution is becoming more serious as more reactors are being built. First results show it's not serious yet, but the work goes on constantly, both on and offshore. At the stern of the ship, John Kreuter prepares to scoop up a sample of the ocean floor.
Well, the first thing we do when we're going to do benthic samples is we like to have some sediment, because we find a lot of the animals are correlated with general sediment distribution patterns. So by using a small box dredge, we're able to achieve 2 things. One, we get our sediment sample, and 2, we test the bottom and see if we want to put our bigger dredge over. If we find that's nothing but rock, we just don't bother putting the bigger dredge over. Sediment samples are taken back and analyzed for grain size analysis, carbonate content, and organic content.
The organic content means any living animals. John is interested in the kinds of animals living on the ocean floor. He wants to know how the living community varies with bottom conditions and the time of year. Since the actual analysis will take place on shore, the samples from this box dredge will be taken below decks and frozen to prevent any organic material from being decomposed by bacteria. The box dredge indicates that the bottom here is not too rocky, and it's safe to put the bigger dredge over the side. This modified anchor dredge is designed to capture larger specimens. It is covered with wire mesh to let sand and mud wash through as it is pulled along the bottom. On the bridge, Jimmy Harker watches as the dredge is maneuvered into position.
6 minutes. 3 to 1 on the depth. You can get it.
As the mate on duty, he must control the speed of the winch as the dredge is lowered to the bottom. By watching the wire angle, he can let out enough line so the dredge will hit the bottom in the right position to dig in.
When the anchor dredge hits the bottom, it digs in to a depth corresponding to the point at which the lip hits the sediment. And when this happens, it stops digging and is pulled through the sediment. The large screening on the back simply sorts out the finer parts of the sediment and keeps larger animals and larger shells, fragments and things like this. Once the samples are brought up, they're preserved, and then we take them back to the laboratory and pick through them, pick out all the animals, and identify the various forms we find. The hope here is to have generalized distribution patterns, both spatial and by sampling seasonally, we hope to get some sort of temporal patterns on the continental shelf.
In an earlier program, we watched several experiments dealing with the lower end of the food chain and the productivity of plant life in seawater. Lowell Sick is also interested in this aspect of marine life. He collects his samples in a net towed behind the ship.
The organisms that we're using are marine copepods. These are microscopic or near microscopic crustaceans. They're nearly at the whim of the currents, water currents or wind-driven currents, but they do possess the power of some swimming ability.
In order to count the copepods, they must first be separated from small debris in the water. It is a tedious process requiring great care.
The organism's significance is that they are the prime link in the food web between the microscopic plant life and converting this plant life to food for higher forms of organisms in the food web, such as larger invertebrates and eventually on up to the fish. Now, the field phase of our project, which we're conducting here on the Eastward, is to look at baseline information regarding these copepods. For example, we're looking at the species that are found in coastal Georgia, the distribution of these species on out to the Gulf Stream, and thirdly, any seasonal variation in both the species composition and their distribution and numbers.
Once the copepods are separated from other microorganisms and trash in the water, the next step is to get a constant number of animals in a given volume, so that the experiments done later with the copepods will be accurate.
And and that means counting out many, many organisms. We have to know how many organisms are in our samples so that we can quantitate what we do. And for a human to sit down and count out several thousand near microscopic organisms is an incredible chore, obviously. So this machine simply counts whatever you want to count. If we want to feed copepods, we can count the number of cells we feed them and get us a specific concentration of food so that this will not be a variable in our experiments.
378.
On deck, Peter Schlein and John Kreuter are putting a trawl net over the side. This is the same type of net used by shrimpers and other fishermen. The wooden skids, called barn doors, will hold the net open and near the bottom as it is towed slowly behind the ship.
Station 6 bridge, we'd like to leave it in the water for about an hour.
While most of the biologists on board are interested in small marine animals or plants, Peter is collecting fish and other larger animals for the aquarium at the Marine Research Institute on Skidaway Island. There they may be used for education as well as research. When it is time for the catch to be hauled aboard, several extra hands are needed. The net must be handled carefully to keep it from tangling so it can be folded and ready for use again later. The catch this time contains a variety of shells, crabs, fish, and other marine life. Of course, Peter is most interested in the fish. He transfers them to a tank with constantly circulating seawater.
All right, these red ones, there are 3 of them right there, are called pearly razor fish. This guy right here with the bluish stripes is a striped sea bass. Over here, these with the vertical stripes are called spade fish. Well, here's a seahorse. This guy down here is called the triggerfish. That's about it. You can't see them. We also have a couple of fat fish in there. There's an octopus in the jar.
Day runs into night. But on Eastward, work goes on.
2 minutes.
Everyone is assigned a duty watch. 4 hours on and 8 hours off around the clock. With the coming of darkness, many animals rise to the ocean's surface and are attracted to Eastward's lights. Along with their other duties, the scientists keep a record of all the living organisms they observe and occasionally use dip nets to bring some interesting specimen aboard for identification.
It's a neat little crab. Is somebody wanting? Put him in the tank. Huh? Okay. This thing bites me. I'm going to jump overboard. How can I put him in the tank? I got him with both hands. Drop him.
This is where we take a shower on the Eastward.
A summertime cruise like this one often encounters a few thunderstorms, but a little rain doesn't slow the pace much.
One more slack on the main wing.
The crew, of course, prefers good weather, for high winds and heavy seas, especially in winter, can make work on deck nearly impossible and very dangerous. Today, though, just calls for rain gear and careful footing on the slippery decks. Some people evidently just like getting wet. In better weather, fire hoses provide cooling saltwater showers. Even the porpoises feel playful today. They get a free ride by swimming close to the bow, where they get pushed along by the ship's waves. And everyone appreciates a chance to rest and soak up a little sunshine. It's been a busy and productive 10 days since the ship left Beaufort. Eastward has now completed the first part of this cruise, and the ship is heading for Cape Fear, where John Newton has a special project to work on. Up in the electronics room, John is studying his charts in preparation for tomorrow's work.
This project started about 3 years ago when Fred Kelly, one of our oceanographic party chiefs, made an accidental discovery. And this is the way things happen in science very often. The ship had just been outfitted with a new sonar transducer, and we were able to, for the first time, get some reflections from below the seafloor from the layers in the Earth's crust, actually beneath the bottom. So Fred saw on some of the first profiles that we had buried channels in the area just off Cape Fear, North Carolina. So this was pretty exciting. The profiles were very evident, they were very crisp and clear, and so since this had never been described before, we thought we would try to put it all together, but it takes a large number of cruises to do this. We'll start by putting the seismic profiler, that is an air gun, in the water, and this just shoots a burst of air into the water at roughly 1 second intervals, and it creates the shock wave. You can see this big bubble occur in the water. The shock wave goes down through the layers of the seafloor and bounces back from each one to the hydrophones, which we trail from the other side of the ship, and then they come through the hydrophone cable to an amplifier, which filters and amplifies the signal, and then it's fed into the precision depth recorder or the seismic recorder so that we could show this on paper.
Well, this is a Raytheon recorder, and normally when the ship is underway, we operate it continuously. The main purpose of it, of course, is to profile the bottom or the ocean floor. The thing does no more than send out a signal through a transducer that's mounted on the hull of the ship. You hear a strong signal, which is the outgoing signal or outgoing ping, and then a return, which is not quite so strong. That on the scale it's operating right now, it's a half-second sweep. So it just measures the distance through the water in a half a second.
Now John is setting up the machine to monitor signals from the hydrophones. He is hoping to see a reflection of the sonar signal that will outline a section of the buried river channel. This previous chart, turned sideways, shows a profile of one such channel.
And this way we'll be able to make a crossing over the buried channels and see just where they are. Then we can swing the ship around and come back in another line, maybe a little bit to the southeast, and make another profile, and we'll keep this up moving toward the southeast all the time until we have enough profiles to tell just where how the channels are shaped and where they're going. The edge of the channels apparently are Miocene outcrops, very hard sandstone. We'll be using a rock dredge, which is just a big steel box with a chain bag on the end of it, and it has two bulldozer blades, one on each side of the steel box. So we can drag the dredge across the channel, and when it hangs up on a rock outcrop, it actually has the same effect as a bulldozer hitting that outcrop. It just breaks off chunks of rock, and they fall into the chain bag, and then they bring this back on board.
The biologists aboard find this first dredge haul quite interesting.
Yeah, some crabs.
Do you want these starfish, Sherlyn?
But the dredge recovered little geologic material.
Yeah, we didn't really hit the outcrop. Let's put her back over and go for about a half hour.
Okay, all right.
Northwesterly course.
The ship is repositioned according to the sonar profiles for a second try. The resistance of the mud and sand on the bottom holds the dredge back, putting a great strain on the towing cable. To keep it from breaking, the cable runs through a set of springs which absorb sudden shocks. The scale shows the strain in thousands of pounds. Sharp tugs on the springs follow one another as the dredge hits something solid on the bottom.
Inside.
Finally, John decides to bring it up for a look.
Right there, Harold. Lower it down a little, Thomas. Yes.
big chunk. This is really an exciting dredge haul. The dredge was completely full of these Miocene sandstones, and you can see that they're pretty well perforated. They've got big holes going through them that apparently were drilled by Pholads, the boring clam. And when we get a chance to open them up a little more and look at them more closely, we'll probably be able to find some fossils that are tens of millions of years old, and also get a better idea of the sediment at the time, that is, the sizes of the grains in the rocks. So this is going to be a big help in understanding the margin of this buried channel, how the banks of the channel are.
Is this mostly from the bank of the channel then? So we were on the edge and not in the center of it?
No, the center of it is filled with all that shelled hash.
Even with this new evidence, more cruises will be needed before John can fully explain the buried channels. Everyone would like to continue, but Eastward is due in Beaufort tomorrow morning. At sunrise, Eastward is approaching Beaufort. Slowly, she sails down the channel toward her slip at the Marine Lab. In a few months, these scientists will be back off the coast of Georgia. The sampling and study will continue in an effort to construct a complete picture of this part of the marine environment and to trace man's actions as he changes that environment. The data collected during the past 10 days will require weeks of study and analysis. Meanwhile, Eastward will lie at her pier in Beaufort Harbor. Her crew will rest, and in a few days, Eastward will put out to sea once again, with a new group of scientists, studying some different aspect of the world's oceans. For that is Eastward's purpose. With each new cruise, she helps the scientists she carries to explore the marine environment, pushing out the boundaries of man's knowledge about the world in which he lives.